Hi, my name is Megan Kelly, and I'm going to be talking with you today about transcription. Um, Before I get started, I just want to talk a little bit about the DNA and RNA, things that you just kind of need to know before you discuss the transcription process. Um, the first thing is that everybody kind of has heard about DNA a little bit, and DNA it looks just like this. And as you can see, DNA has two sides to it. It's like a double helix. And with DNA being a double helix, it's basically like a ladder. So, and the things connecting it in the middle are bases. Those bases are called cytosine, guanine, thymine, and adenine. And there's one thing to remember with this is that they have to be paired in a certain way. There's a kind of a rule to that we'll talk about later. Um, Basically, always cytosine and guanine is always going to be paired together, and thymine and adenine will always be paired together in DNA. The purpose of DNA is that it stores and transfers genetic information. So basically, everything about you, from your eye color to your hair color, everything is stored in your genetic information inside your DNA. Everybody has kind of heard of DNA before. It's kind of common knowledge that DNA is the reason that you're the way that you are. But I just wanted to go ahead and talk about DNA so that we could talk about RNA, which is not quite as heard of among certain people. Um, RNA looks kind of like DNA, but it's missing a letter. Um, it just has one helix, and as you can see, there's only one side that has the base pairs on it. It's just one strand is what RNA is. The purpose of RNA is that it codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. So there's your picture of RNA. There's one difference in the base pairs, because as you can see, there's also still base pairs. There is guanine and cytosine, which is just like in the DNA, double helix, and those will always pair together, just like in the DNA. But also there's adenine, and instead of thymine, there's uracil. And that's where the adenine will always pair with uracil when it comes to RNA. So this is what RNA looks like. Okay. Now for the base pairing rule that I talked about earlier, here is a little bit of that. It's basically saying cytosine will always pair with guanine, and adenine will always pair with thymine, or adenine will always pair with uracil. It's pretty easy to remember, but some, I mean, it might not be for some people, but it's just simple as that. Those always pair together. They don't switch up. They don't mix up. This is how they'll always be paired. So now we're going to talk about transcription. We're going to talk about transcription. Basically, what transcription is where it turns, basically DNA turns into messenger RNA. And so see, there's RNA right there, and the M stands for messenger. And I'm going to tell you how that happens. Now, DNA, it contains your genetic information, and the messenger RNA, the purpose of the messenger RNA is that it carries the information to the other parts of your body, to the ribosomes, to where other DNA can be made, or can get your information from that as well. So here's that. So here is how DNA is turned into messenger RNA. You start out with the DNA up here, and I know this is probably backwards, and I don't know really how to turn that around, but okay. This is my strand of DNA right here. As you can see, it's got the two different blues. Okay, it's just regular old DNA. Well, DNA, when it starts to turn into messenger RNA, it kind of pulls apart. As you can see right here, the helixes kind of start to spread. Whenever that happens, there's different bases that are floating. They're floating around around the DNA, and as this spreads apart, this orange line right here are bases that come, and think about it like magnets. They just suck in to that part because they think that it needs a pair, because whenever the helix splits up, the pairs that are in the middle, they split apart as well, and the bases that are floating around think that there's openings and that they need to go be paired with that side of the double helix. So as that happens, as this pulls apart, it's kind of tricking them. It pulls apart, and the pairs pair up right here and create this little piece right here that lifted up. That's called the sensory strand, and so basically that strand gets copied. So this little orange thing right here is basically the same exact thing as what lifted up. And so once this is finished and it's a long enough piece, there's enough pairs, it starts to rewind. So as you can see, this strand starts to come back down. And so it looks like regular DNA again. And then this piece right here that was copied, that was the pre-messenger RNA, gets dropped out right here. It gets dropped out. And so basically all that strand is that it is a copied piece of the sensory strand of the DNA. And so that's how your information gets taken and copied and put into other strands of DNA and other parts, turns into proteins in other parts of your body. And so that the next step, which is the very last step that I didn't draw a picture for, but this little yellow orange strand right here, the pre-messenger RNA gets taken and spliced into little pieces. And whenever you think about it being spliced, just think about it being cut up into smaller pieces, just like you would cut up some food or some vegetables or something. It's spliced into smaller pieces, and it's taken and it is paired up with other strands. That way they make the base pairs. So basically that's what happens with that. And the next step would be the transcribing. And my friend Martha is going to talk to you about that. So thank you for your time, and I'm really sorry if this wasn't informative to y'all. Sorry.